非常高兴的日子，因为我们说实话，刘福东刘院士是今天我们的讲者。那我们历经了几年的 COVID-19 的这个 outbreak， 所以过去这几年都是刘院士利用线上的方式来做演讲，以及对于我们学校老师跟同学们的指导都是靠线上。那好不容易我们现在整个疫情趋缓，然后刘院士现在也是旅居美国为主哈。那在这一次百忙之中有回到台湾，那我们也正好这个天时地利人和全部都在一起，然后才有办法邀请我们的刘院士来这边跟我们一起见个面。那我想今天最主要的讲题还是由我们刘院士来进行他对于我们的有关于 glycoprotein 的这些 research 有哪些比较新颖的一些发现。那我们也会希望我们在座的不管是师长或线上的师长同学们之后在刘院士报告完之后分享完之后，再跟我们一起跟刘院士请教。那我想我们就借各位的双手欢迎一下我们刘院士的莅临。
谢谢。
有了，是不是？
好，谢谢谢副院长、教务长的介绍，还有院长的 invitation。那确实是非常高兴，再次到慈济，多年来疫情的关系，没有人到实体来演讲，这次是几年之后第一次 in person 的 visit， 然后跟大家分享一下我的研究。是用英文，对，用英文，对。So I started to speak in English. So my research has been for many years on galactans, which I talk about quite a bit in the previous visit or online presentation. Galactans are glycan-binding proteins, and for this presentation, I thought I would cover a broader topic. That is glycan-binding proteins. So this only about 10 years, a little bit over 10 years ago, National Research Council of the National Academies, this is in the U.S., including National Academy of Science, National Academy of Medicine, National Academy of Engineering, they published a document entitled Transforming Glycoscience, a Roadmap for the Future. And they stated that it can make contributions toward understanding and improving human health. They emphasized that now is the time for glycoscience to be embraced broadly by the research community. And although these challenges are substantial and complex, the results of achieving these goals have the potential to impact science in exciting ways. Now, a few years later, in Nature, there's a short article entitled Sweet Success. And biologists are diving into sugar molecule research thanks to new tools and techniques. By new tools, mostly is in glycomics. So we have genomics, proteomics, and there's glycomics. And glycomics has always been behind the other two, omics. But it's catching up. Actually catching up pretty quickly. So glycomics is the systemic study of all glycan structures of a given cell type or organism. By glycan, actually this slide covers all the glycans that you need to know, including all linked glycans, which is attached to serine or threonine, and the unlinked glycan, which is attached to asparagine. And glycosphingolipid, which is glycolipid, and then proteoglycan, which has a protein backbone, and then glucosaminoglycan branches. Inside the cell, there's a simpler glycosylation that is a monosaccharide, N-acetylglucosamine, also linked to serine threonine. In fact, the size that is modified in this intracellular glycoprotein are the same size, or some overlap, some do not, by phosphorylation. So the sites that you're familiar with, phosphorylation, they can be glycosylated as well, but the only monosaccharide is the O-linked N-acetylglucosamine. It's a very important discovery in terms of the intracellular glycosylation. So for this presentation, for the purpose of this presentation, I know that these glycans are kind of a Greek word to you, and you're not necessarily familiar with them, but for the purpose of this presentation, you really do not need to worry too much about N-glycan, O-glycan, and branching structure, and so on, and just think that glycan as a pattern recognized by glycan-binding proteins, right? Yeah. So then you find that this actually may not be too difficult to understand what I'm trying to convey to you. So it's good timing to show this protein, this glycoprotein from COVID, SARS-CoV-2. So SARS-CoV-2, I think this is RBD. So the purple, green, and blue colored area are glycans. And then you can see this protein heavily glycosylated. We call this glycan as a glycan shield. I used to present also HIV, GP120.
And we need to stop because
Okay.
the
Not moving.
Yeah,
Not moving. Yeah.
手动枪。
No. Do we need to do this?
Can we go to the full screen?
Moving? It's okay now? Okay. All right. Okay, sorry about that. It's technical issues. So maybe I just start over with this slide.
Excuse me.
Okay. So this is SARS-CoV-2, RBD. This is glycosylated glycoprotein. And those color purple, blue, and green are glycans. You see these glycoprotein are heavily glycosylated. And we call this glycan glycan shield. I used to present HIV, GP120. There, the glycosylation is even more dramatic. It's pretty much is totally covered by glycan. You can hardly see any peptide backbones. So actually, for all of us have been vaccinated using if it's mRNA, then our body will take care of glycosylation. So the protein that you are eventually exposed to, glycoproteins that are also heavily glycosylated. So you will make antibody response to those glycan as well. It's possible. So those are quite complicated glycoprotein, but the IgG that you're more familiar with is simpler. And there is only one glycosylation site that asparagine. This is N-glycan, only one. But even there, you see that there's a glycoform, a different glycoform. For example, you might be familiar with biologics. That is, these days we use monoclonal antibodies against various mediators like TNF for treating diseases. And there, for example, one is Humira, which is anti-TNF, human necrosis factor, and there's 50 also glycoforms. It's the same protein backbone, but the glycan structure can be variable, and what the patients received contain maybe 50 or 60 or so different glycans. And I didn't know this earlier, but these various glycoforms, they might have different functions. For example, some can be pro-inflammatory. IgG glycosylated in certain way may be pro-inflammatory, but they are glycosylated in different way. For example, they're more heavily silated. They can be anti-inflammatory. Then these different glycans have different impact on the function of antibodies or IgG. For example, for antibody-dependent cellular cytotoxicity, ADCC, it's important to know that the fucus is actually the presence of fucus in this position is inhibitory of ADCC. Because this IgG will bind to Fc receptors.
We need to redo it again. Excuse me. 不好意思。
用成那个。
So we cannot use the.
整个荧幕对，用这个。然后测试一下。
不见了。这里可以。
对。等一下。
要试一下，要试一下。
对，要测试一下。
可以吗？
他一定会累的。
对，他会累的，稍等一下。
Okay. Sorry again. Okay. So maybe I repeat this slide. So I mentioned in the previous slide that IgG has only one glycosylation site, but even there are 50 or more glycoforms, and the variable glycans can have impact on the function of IgG, even though they have same protein structures. And one very well-known variable is fucose. So if at this position, if the antibody is fucosylated, then it can have negative effect on ADCC, that's antibody-dependent cellular cytotoxicities. So a pharmaceutical company generating antibodies or monoclonal antibodies as biologics for treating diseases, they need to remove fucose if the antibody is going to be used for, or this biologic is going to be used for treating, say, a tumor using the mechanism of antibody-dependent cellular cytotoxicity. The reason for the impact of fucose is that antibody bind to IgG receptor, Fc gamma receptor, and there this glycan is not directly at the binding site, but it's close to the binding site that bind peptides. And in the presence of fucose, the areas will be a little bit crowded, therefore it will be low affinity. And if you remove fucose, it will become high affinity. Actually, we're talking about like 50 times difference in terms of the ADCC activity, so it's quite substantial. In terms of glycomics, for very simple IgG glycosylation, now you can have automated high throughput IgG antibody glycophiling platform. So very small amount of protein, you can determine the glycoforms. And interestingly, just use very small amount of serum. You can actually, without purifying IgG, you can determine the glycosylation by the high throughput glycomics platform. And these authors, the senior author is a Golden Log from Croatia, and they published and stating that published papers and stating that end glycosylation of IgG is altered in autoimmune diseases, various other diseases, and has potential to serve as a prognostic or diagnostic biomarker, and can complement traditional clinical tests. He stated in his lectures, I heard a few times, that in fact, if you analyze IgG glycosylation, might serve as a reveal your biologic age, not chronological age. And he stated that if you are healthier, you have one kind of glycosylation. If you may not be as healthy, you can have different kind of glycosylation, but this can be reversed meaning that if, for example, you engage more exercise or so on, then the glycophone can change your biological age. I thought it was quite interesting. So I'm looking forward to the development in this regard in the future. So there's a lot known about glycosylation in cancer. So cancer cells in general have high fucosylation and sialation. And there are a number of glycans now have been shown to serve as biomarkers for cancer. And I circled a few. These are, I believe, mostly, I know it's in fine print, so I will not go into detail. Just saying that these days, through very powerful glycomics, you can identify glycans that are uniquely expressed in cancer cells, not in normal cells. This may be useful for treatment using either antibody to target cancer cells or for the purpose of vaccination. Now, those are glycomics, but this term functional glycomics. So those who thought we need the term functional glycomics stated that to decipher the function of glycan, we need to identify proteins that recognize the glycans. They can translate the information encoded by specific. Yeah, I think we got into trouble again.
The mouse will appear in the big screen, not in the here.
Yeah, but we're not being able to advance the slides. Okay, now it's good.
Okay.
Yeah, sorry about the technical issue. Okay, so to decipher the functions, we need to identify proteins that recognize the glycans that can translate the information encoded by specific glycan structure. So functional glycomics is to define the concerted and regulated action of carbohydrate binding proteins, that's glycan-binding proteins, which is the major topic of today's presentation. Okay, no, so it's not synchronized. Yeah.
所以可能变成说要它可能切两，对。
不是因为你要切什么？要让我？
因为
那就是
对，因为它这个它的 PPT 没办法，这跟这个没同步，所以。 等一下哦。解决一下。姐姐帮我看一下那边有没有在动，有吗？有吗？好。
Okay. Okay, looks like it's working. Yeah, it's working now. Okay. All right. Okay. I think that's okay. So previous about 10 minutes just introduction of a glycan part. Now it's coming the more major aspect of my presentation. That's the glycan binding proteins. So there are the slide showed major types of glycan-binding proteins. So I think you're familiar with selectin, which is a very well-known cell adhesion protein. Yeah, I'm going to use my. And then the C is a C-type lectin. There are numerous. I'm talking about maybe 100 of different C-type lectin-like receptor. You might have heard the presentation by Professor Xie Siliang, and he works with several different C-type lectins, like CLEC5, CLEC2, and so on. And there's so I will talk about selectin a little bit, and also I-type is a CGLEC. CGLEC is a sialic acid binding immunoglobulin, contain immunoglobulin-like structure, and I will talk about this a little bit too. So galactins are quite different. They do not have transmembrane proteins, transmembrane domain, and they do not have so-called signal sequence or peptide leader sequence. So they're present mostly inside the cell. However, they can be secreted. And so this is this makes the study of galactin a little bit complicated, because the glycan they bind to mostly outside the cells, just like I show in the previous slides of various type of glycans. But they are galactins inside the cell. So what's going on there? And so I would actually my talk will be mainly focused on intracellular function of galactins. It turns out you can have a glycan independent function and a glycan dependent function. So this is actually to me, I just made these slides not too long ago. I think it's actually, to me, this is probably the most important slide today. I can spend a little bit more time on this one. I hope it's very clear in terms of the message I'm delivering, because this is very troublesome in terms of studying glycan-binding proteins. And so I believe that researchers working with glycan-binding proteins and also readers probably need to be aware of these challenges. Okay. So you're all familiar with cytokines and chemokines, and for any given cytokine or chemokine that discovered, a few years later you will discover is their receptor. Now it's always just one cytokine is one receptor, one chemokine become a receptor. Then pretty much the rest is up to functional studies, and you don't need to worry about what receptor actually responsible for what kind of function. So very clear cut. Of course, I'm not trivializing the discovery of function, but on the other hand, you don't need to worry about how, because it's bind to specific receptor. But glycan-binding protein, they bind glycan, and this given glycan-binding protein may engage various glycoproteins. Different glycoprotein may carry the same glycan ligands. Right? So, for example, I will talk about galactins. They bind galactose, and so various oligosaccharides they contain galactose can be recognized by galactins. But those glycans are present on various different kind of glycoproteins, just not one. So identifying one glycoprotein that's recognized by one type of glycan-binding protein, does not help understanding the function, right? So even you find many might not help, because so that's the challenging. And I hope this will become more clear later. And I hope you ask questions. And actually, recently I wrote a review articles. It took us, me and my co-author, Sean Stoll, he's a professor at Harvard, almost like 3 years to complete this review by publishing Nature Review Immunology. And we address these issues, hopefully quite clearly in the review. So selectins, I think you're familiar with, so I won't spend too much time. So there's just three. Other glycan-binding protein have many family members, but it's good that selectin, there's just three, L, E, and P. And these are very important cell adhesion proteins. They control leukocyte trafficking. Because of the timing issue, so I'm going to skip a couple slides here. So this is already very classic biology, and it's probably in many biology textbook. So selectin, when earlier known to be protein recognized by certain monoclonal antibodies, and then when the protein structure was what the monoclonal antibody binds to was elucidated, then recognize this is the lectin, that is carbohydrate binding protein. So the name of selectin was given. And so those are very important development in biology. Then what they bind to was revealed, that is silyl-Luex. Again, this is a very important progress. Then scientists started to try to identify the glycoproteins they recognize. So PSGO1 came out. This is a P-selectin glycoprotein ligand of selectin. And then it turned out this is recognized by L-selectin, P-selectin, E-selectin. Then there are a number of others. This is a really very, very partial list. I didn't spend time to add more. But they are, for each selectin, there may be numerous glycoprotein recognized. So these days, I don't think selectin has been known for over 30 years. So I don't think these days people worry about what glycoprotein selectin bind to. In fact, we do not know what whether the function of PSGO1 is necessarily related to selectin. So if you somehow block the PSGO1 or you engage PSGO1, does not mean that you because you affect the selectin PSGO1 interaction. Okay, so this is just the one explanation for what I mentioned to you about in terms of challenges. It's good news that for all the glycan-binding proteins I talk about, they already drugs that have been developed. That's, so my title is therapeutic implications. So this took over 15 years. So P-selectin is known, maybe I would say that 30 years ago, but the company started, and this company wanted to develop a drug against P-selectin, and took quite a few years for them to decide they want to target sickle cell, and because P-selectin not just bind to leukocytes but also bind erythrocytes. They are silyl-Luex on erythrocytes, and as you know, sickle cell anemia, they have a major issue with sickle cell anemia patients is the pain crisis because of vaso-occlusive issues, and this we call vaso-occlusive crisis. And it turned out these antibodies against P-selectin can block interaction between P-selectin endothelial cells and the erythrocytes, therefore relieve the vaso-occlusive pain. And this was became FDA approved in 2019, about 15 years after this small biotech company started. And this is actually was the company was acquired by Novartis. So this is actually a success story, but also indicate that this really takes a lot of time, a long time to develop drug. And this SIGLAC. SIGLAC is sialic acid binding immunoglobulin-like lectins. And the sialic acid, if you do not know, is a monosaccharide, but it has a 9 carbon instead of 6. And sialic acid is just the one term to cover many. Maybe I often ask how many, but maybe 30, 40, or even more sialic acid derivative. It's not just one sugar. It's not, unlike glucose, just one structure, but also you can have glucosamine and so on. So sialic acid is just a general term to represent a group of 9 carbon sugar. So that's one domain binds sialic acid, and the other domains has a sequence homology with immunoglobulin. In cytoplasmic tail, there's a immunoreceptor tyrosine-based inhibitory motif, ITIM. As ITIM is activating, and the ITIM is inhibitory. Indicating or suggesting that these proteins may be involved in downregulation of intracellular signaling pathways. There are now 16. This slide is a little bit outdated. It's only 15, but there are 16 SIGLAC now being recognized. They are conserved ones, meaning that they remain quite homologous through evolution, but there are some more rapidly evolved Siglecs, designated as CD33 related Siglecs. And they evolve so much so those in mice may not, it's hard to determine whether they are corresponding to which kind of which Siglec in human. Siglecs are mostly studied in leukocytes. So all leukocytes contain 1 or 2 or 3 or more Siglecs. As mentioned, that they have IT motif. Most of them have IT motif. This is represented by these red circles. So on cell surface, if some glycoprotein or protein engaged, they can generate activation signals. But if 1 or 2 or more of the Siglecs are engaged, then the inhibitory signal can be generated because of IT motif and leading to suppression of this activation signal. Okay, that's in general how Siglec function. So in terms of therapeutic implication, so NK cells express one of a Siglec called Siglec-7, and as I mentioned, the Siglec binds salicylic acid. I also mentioned that in cancer cells have more, in general, more highly fucosylated and highly sialylated. So on the surface, they are multiple glycoprotein that have salicylic acid in the glycan, and they can engage Siglec. By doing so, interaction of salicylic acid on tumors, it's on the right-hand side, tumor cells, there's a typo there, and the Siglec NK cells may dampen NK cell activation in the tumor context. So NK cells now is inhibited by tumor cells. So Siglec might be considered as an immune checkpoint inhibitor. I think you're familiar with PD-1, PD-L1. This is the Nobel Prize that took a couple of years ago. And this is the immune checkpoint pair. So when PD-1 is engaged on T cells, then T cell is suppressed. So we want to block PD-1, PD-L1 interaction, then the T cell can be more active. Similarly, I believe that if you want to block Siglec-7 and salicylic acid interaction, then NK cell can be reactivated and might be able to overcome this tumor effect and also kill tumor cells. So I think this antibody against or blocking Siglec-7 should be now undergoing clinical trial, although I don't have information on that. Okay. Another therapeutic implication is Siglec-8. Siglec-8 is expressed by eosinophils and the mast cells. So Bruce Buckner, he used to be professor at Johns Hopkins. He moved to Northwestern, and he discovered that when you engage Siglec-8 is delivers suppressive signal, right? But it turned out it kills eosinophils, and this might have some implication in the resolution of eosinophilic inflammation. So on the right hand side. So it shows that when maybe some eosinophilic inflammation, subsequently the Siglec-8 get engaged, and eosinophil would die, and so this eosinophil will disappear. And he followed up with discovery in developing antibodies. In this case, it's agonistic antibody. So anti-Siglec-8 antibody turned out to be able to kill eosinophil, and he led this to a clinical trial, and subsequently the company developed monoclonal antibodies against Siglec-8. So the name is lirentelimab. It's now FDA approved for treatment of eosinophilic gastritis and duodenitis. So another success story in glycan-binding protein field. And I understand that clinical trials for urticaria, which is very common skin issues, and the systemic mastocytosis are both being conducted. I've seen the result, and it looks like they are promising. So this will be a big news when they get FDA approved for treatment of this disease. Now galactans. So this will be the topic of remainder of my presentation. So I hope I still have time to cover this part. So for us, our engagement of galactans was by accident. So my group was interested in IgE and IgE binding protein, Fc receptor, IgE receptor actually on mast cells, basophils. And back then, cloning takes some effort. These days you don't need to clone because all the sequence are already is known because the whole genome is sequenced. So back then we wanted to clone IgE receptor on mast cell. We end up finding another protein that also binds IgE. We call it IgE binding protein. We didn't know until later that this protein is actually also lectin. Lectin is glycan-binding protein. Okay? And the carbohydrate that the IgE binding protein binds to, all IgE binding protein binds to is galactose or lactose. And then there are two homologs in the literature, IL-14, IL-16. It actually turned out they are another one. So those of us working on this homologous protein decided we need to give a name to these proteins. So we give name galactans. So these are lactans, just like selectin is a lactin. Siglec is a sialic acid binding immunoglobulin like lactans, right? So galactans, so galactose binding lactans, a family of animal beta galactoside binding lactans. And so we send a note to editor, published in Cell. Just recently, I discovered that actually now this letter, just one page letter, has been cited as over 1,600 times. And in fact, for galactans, so this is 1994, so it's already, time flies, it's already 30 years ago. So if you do a keyword search for galactan in PubMed, you'll find close to 11,000 publications. So this has become a pretty big field. And our IgE binding protein now is known as galactan-3. So we don't refer our protein as IgE binding protein. So we kind of want to forget about that history. But this is galactan-3. So galactans is beta galactoside binding animal lactans with conserved CRD, carbohydrate regulation domain CRD. It's evolutionarily highly conserved. It's unlike Siglec, which is evolutionarily highly not conserved. So between mice and human, you hardly find the sequence homology. But this is different from for galactan. And so there's mainly three types. One is protype. They have only one CRD. They often exist as homodimers. And two CRD, the homologous CRD linked by short peptides. Galactan-3 is what we are first to clone in sequence, and this IgE used to call IgE binding proteins, which has a CRD and a non-lectin area. So some galactans have wide tissue distribution, others have high tissue specificity. I'll give you examples. And I already mentioned that galactans are different from other glycan-binding proteins in that they do not have transmembrane domain. I also mentioned that they do not have a classical signal sequence or leader sequence. These are the sequences you need for a protein to be made and then subsequently secreted. So consistent with this, galactans are found mainly in the cytosol. They can go to nucleus. They can go in and out of nucleus between nucleus and cytosol, but also in extracellular space. There are quite a few publications about how they might be secreted, but still it's not maybe quite a few different pathways for them to be secreted. Like lectin, and lectin in Chinese is ninjisu. So related to that, they often bivalent or multivalent. And so this applies to galactan as well with regard to carbohydrate binding activities. You might find that what I will talk about is in terms of biology of glycans are pretty complicated, which is true. But it's good news is for today's title, therapeutic implications. Many glycans have been implicated in the pathogenesis of a variety of diseases. I'll give you a few examples. So I put a question mark here in terms of extracellular function. This would be a good topic to discuss. I hope you ask me questions later on for me to clarify it. But I mainly talk about intracellular functions. So this includes glycan independent functions for 3, 7, 8, and 12. And but I think a very exciting development in the field. It's almost like 30 years after discovery of this family, the glycan dependent function inside the cell. Right. So I will share with you toward the end of the presentation. So glycan-3, I mentioned that it has a CRD and the non-lactin area. It can form oligomers upon binding to multivalent carbohydrate and has wide tissue distribution. Almost every tissue you know of either can make glycan-3 or can be induced. And like other glycans, glycan-3 is present in cytosol and nucleus. We thought early on, including our own group, we tried to find the function of glycans. We just added a recombinant protein to cells and see what happened. And we say then we code this as a function of glycans. But quickly we thought this really might not be a good approach because we should ask the function of endogenous glycans. Endogenous meaning 内源性, that is what the cell comes with. It's not something you add from outside. Okay. And then together with that question, we ask whether they have intracellular function because they are present inside the cell, right? And it's very lucky for us, but this is really a lot of hard work. We discovered the number of function for endogenous glycan-3, and those are intracellular function. I won't go through this slide function one by one, but just to show on the left lower corner is apoptosis. So this is the very first function of endogenous glycan-3 that we discovered. And we also found that glycan-3 interact with BCL2. BCL2 is a well-established anti-apoptosis protein, and glycan-3 interact with it. It turned out that glycan-3 has some sequence homology with BCL2, almost like it's a very remote relative of BCL2. So it makes some sense it has engaged anti-apoptosis activity. So the interaction with these intracellular regulators is how glycan-3 function and how endogenous glycan-3 function. And these function is independent of glycan binding because BCL2 is not glycoprotein. And another protein we discovered that glycan-3 interact with is on the upper side of the slide, the ALEX. ALEX is becoming, I think when we published it, it was very early on. ALEX is not well known. But now ALEX is very well known to be involved in protein trafficking. And it's not a glycoprotein. The glycan-3 bind ALEX bind very well. So we cannot exclude that carbohydrate binding site may be involved in interacting with this protein. In terms of ALEX, I think it's the N-terminal portion, non-lectin part that's interact with ALEX. So TRD is actually not involved. So over the years, we and others found that the glycan-3 in general is a positive regulator of a variety of inflammatory responses and thus a variety of inflammatory diseases. There are exceptions to this, meaning that there may be negative regulators, but in general they are positive regulators. Interestingly, in a heart failure situation, glycan-3 is now shown to be a serum biomarker. And for someone with a symptom of heart failure, you can look at the serum glycan-3 level. If it's higher, maybe indicate that the prognosis is not good. If it's lower, then it's have good prognosis. And so glycan-3 is actually now pretty well known among cardiologists. And so It turns out, I think, just Glycan-3 alone might not be sufficient to make accurate diagnosis or prognosis, but maybe but together with other markers might be quite useful for managing patients with chronic heart failure. And there's huge literature on Glycan and cancer. And more than 15 years ago, I wrote a review on Glycan and cancer, but these days it will be very hard to write the reviews for so many papers. Maybe on one given Glycan may be possible for Glycan-3 or maybe Glycan-9, for example. Okay. So therefore, my conclusion is, or in general, we conclude that Glycan-3 may be targeted for treatment of diseases. Some studies suggest that Glycan-3 functions intracellularly, while others suggest it functions extracellularly. But I must say that those suggesting that Glycan-3 function extracellularly might have used Glycan-3 added exogenously to cells, right, including our early work. So if you look at this publications based on either knockout or knockdown, in general, I think the conclusion is that they work inside the cell. So in terms of therapeutic implication, the big question will be, should we target intracellular or extracellular Glycans? But of course, if you target intracellular, for example, if you siRNA to not allow Glycan-3 to be produced, then you take care of both issues. Actually, there's already a drug they're targeting Glycan-3. This is by a company developed by university in Sweden, Lund University. But I believe the company is in Denmark, so called Galacto Biotech. And they developed small molecule inhibitors targeting Glycan-3. And this has received the US and the EU orphan drug designations in 2020. For the compound, they call the TD139, but now as a drug, they call GBO or GB1, 0139. And these are disaccharide derivative, because glycans bind the lactose with disaccharide. So this is only difference is there's a thiol group, sulfur in the middle. So this thiol diglycoside derivative. So I often ask the company whether this drug target whether this is effective because this is target intracellular glycans or extracellular. And the answer say that they believe extracellular, but it's good for them because this drug is cell permeable, so it's getting inside the cell. So I ask whether if they would have say this company developed by a chemist, so they start with making small molecule drugs. But if they are started by immunologists, they might start with monoclonal antibodies. The question is whether it will work or not. In fact, we have neutralizing monoclonal antibody for Glycan-3, but in our lab, for various animal models, they never show any effect. However, recently there's a paper in Nature Communications. The author developed, I think it's also from a company, monoclonal antibodies against Glycan-3, and they show that they are effective in treating in animal model of systemic sclerosis, that they show the antibody to be effective. However, I think there might be some issues, and so we have to see how this company developed this as a drug. So it's good thing to watch and follow up. Okay, so next slide. Next Glycan is Glycan-7. So this is expressed by stratified epithelial cells. It's highly induced by p53. It's pro-apoptotic. So Glycan-3 is anti-apoptotic and Glycan-7 is pro-apoptotic. So we show that the Glycan-3 is actually a tumor suppressor. And others show that Glycan-7 bind the BCL2. So two Glycans, one is anti-apoptotic, the other is pro-apoptotic, and both bind BCL2. On the right side, we just show that Glycan-7 is highly enriched in nuclei. So it's a clearly intracellular protein, and so actually Glycan-7's field is a little bit I call pure because we suddenly see researchers use recombinant Glycan-7, add to cells and see what happens and correlate the function. But mostly they study endogenous Glycan-7 function. So in our lab, we because I'm a dermatologist, so we focused on one skin disease, psoriasis. Psoriasis is 肝癣. I think you're probably familiar with this. So it's a pretty common chronic skin disease affecting about 2% of population. In Taiwan, maybe we can ask Dr. Zhang. The prevalence I read is a lot smaller, like 0.2 or 0.5, but it maybe it's underdiagnosed. It's a chronic inflammation, maybe it's autoimmune or autoreactive disease. The two features of this disease is hyperproliferation of epidermis and there's inflammatory infiltrate in the skin. Dendritic cells and possibly macrophages are key players. Psoriasis used to be considered as Th1. You know how we have Th1, Th2, allergic disease Th2, and there's Th17, newer T cell polarization Th17. Now has become clear that psoriasis is a Th17 mediated disease. And importantly, psoriasis is a metabolic syndrome, considered as a metabolic syndrome. Patients often have this lipidemia. The reason we got interested in Glycan-7 in psoriasis is many years ago, we did transcriptomic analysis of skin diseases, including atopic dermatitis, AD, contact dermatitis, CD, and psoriasis. This is a collaboration with Genentech, biotech company. So for each disease, we have 13, 15 cases. And we do a microRNA analysis. Eventually, we published a paper. We found that many genes are upregulated or downregulating in each of these skin disease. Then we retrieve the profile for all galactans and found something, several very interesting profile. For Glycan-7 for psoriasis, it looks like in lesional skin, its level is lower compared to non-lesional site. And we confirm this by immunohistochemistry, and the upper panel is non-lesion, lower panel is lesional skin, and you see a lower expression in lesional skin. So this is the now still quite so this is. It's over 10 years ago, but it's still quite accurate in terms of the cellular and molecular mechanism for psoriasis. So you have Th17 and maybe Th1 as well, and this is very cytokine lead to keratinocyte proliferation, and the keratinocyte in turn activate some inflammatory cells such as neutrophils. So our question was, what happened to keratinocyte when Glycan-7 is downregulated? By the way, in skin, only keratinocytes express Glycan-7. So we actually published two papers separately over a few years. So this more recent one in Journal Clinical Investigation, this is the graphic abstract of that paper. I think it's pretty detailed, so I won't go through these slides, but just use the next one to summarize our finding. So downregulation of Glycan-7 in keratinocytes may contribute to the heightened keratinocyte proliferation and the inflammatory response in psoriasis. So we know I mentioned that Glycan-7 is downregulated in psoriasis, and that we show that it contributes to cell proliferation and also inflammatory response. Therefore, we propose that approaches to induce Glycan-7 in skin may be developed as a new therapy for psoriasis. So we're lucky that by really pretty straightforward approach called the connectivity map analysis, CMAP analysis, we found that a number of compounds, including statins, can induce Glycan-7 expression in keratinocytes. therefore, we propose that statins may be employed for treatment of psoriasis, and in the meantime, control dyslipidemia. So I mentioned earlier that psoriasis is considered as a metabolic syndrome, and the patient often have dyslipidemia. So they should take statin to control dyslipidemia, to protect their heart, because they have risk in developing cardiovascular issues. And the statin in turn can also control skin lesions. So I hope this can lead to clinical trials. In fact, I talked to quite a few dermatologists. Actually, statin has already been used for psoriasis. So I'm going to skip 8. It's also on psoriasis. As you see on the left side, in psoriasis, it's highly upregulated. So Glycan-7 is downregulated, but Glycan-8 is highly upregulated. And so we follow up on that, and to show that it contributes to epidermal proliferation. And so the next one is Glycan-12. Again, I'm going to go through this very quickly because I really want to go to the last part of the talk. It's very exciting. And so Glycan-12 has 2 domains, so 2 CRD. And so the one challenge in our lab is we often need to learn something that is now expertise. So this one, Glycan-12, is mainly expressed by adipocytes. So we have to become expert. Of course, we collaborate with experts, but we also need to learn about adipocytes and Glycan-12. So we generate knockout mice and show that these knockout mice have a lower amount of fat, and they can handle weight-associated insulin resistance and glucose intolerance better. It turns out that they are suppressed lipolysis and localized in lipid droplets. So I'm sorry, I'm skipping this. And then as a dermatologist, we work on in the past few years on sebaceous glands. Sebaceous glands are known to have a fat lobule. It's very rich in fat. So Glycan-12 is expressed there. And so knockout mice we have, which we studied adipocytes, we show that they also have smaller sebaceous glands. Okay. And then we follow up on that and have 3 papers related to Glycan-12 and sebaceous glands. So this is one area we're still still working on. I'm already actually at the USC where I work now. And so here we believe Glycan-12 can be targeted for treating skin disease associated with sebaceous gland dysfunction. So I share with you intracellular function of glycans for various glycans, and these so far turn out to be involve protein-protein interactions. So this is not quite satisfactory, although we believe those publications are impactful. But we kept asking whether cytosolic glycans function by binding to glycans. And so by glycan, I meant this I showed you earlier: the glycoprotein, glycolipid, proteoglycan. Right? So these glycans, glycoconjugates, we call, the synthesis started in the ER, and subsequently goes to Golgi, and then they can go to extracellular space. They can be internalized, but all these times, these glycoconjugates are in the lumen of these organelles, whereas glycans in the cytosol. So they cannot see each other. Glycan cannot penetrate through this membrane to get in touch with the glycoprotein ligands, right? However, I didn't know. I wish I knew this. This is not something new. This is people know about this for years. I just didn't know that these organelles can be damaged. There are many disruptive materials. I show you a few can get inside the cell, and this endosome or lysosome can be damaged. And then these glycoconjugates that initially reside in the lumen of these organelles can become exposed to cytosol. Then the glycans in the cytosol, they quickly recognize this glycan. They become exposed to cytosol, and they lead to some downstream function, like a downstream event, for example, autophagy. So unfortunately, I probably need to skip this slide about autophagy, which I hope you're familiar with. And so autophagy is implicated in in infectious disease, as I have this degrade in invading intracellular microorganisms. So we studied this about over 10 years ago, where I was in UC Davis. So Listeria can invade macrophages, or macrophage can engulf or phagocytose Listeria. But Listeria can escape from phagosome. They get into cytosol, and so the phagosome get damaged. And this slide just shows that galectin-3 can accumulate around Listeria when infected in cells infected by Listeria. Very striking localization. Then a very important paper came out. This is from UK. So this author study Salmonella, not Listeria, but Salmonella also get into macrophages, and then reside in phagosome. But they can escape from phagosome, and the phagosome lose integrity, membrane integrity. And so Listeria, Salmonella get out into the cytosol. But then they show that the 3 galactins, 3, 8, and 9, bind damaged phagosomes. And one of them, galactin 8, also bind the autophagy adapter NDP52. And so then galactin 8 target the damaged vesicle for autophagy to defend cells against bacterial invasion. So this is a very, very striking, very exciting development published in Nature. Now it's already cited over 1,000 times. It's a very important discovery in our field. So we had to catch up, and we found in Listeria, galectin-3 is actually suppressed autophagy. We didn't compare, we didn't look at Salmonella, and they didn't look at Listeria. So we do not know whether in each infection this is still true or not. But the galectin-3, anyway, 3 and 8, they both can recognize glycan exposed to cytosol, and then can modulate autophagy. Then why is important? Why is it's exciting? But because many substances can cause endosome lysosome damage. I mentioned intracellular bacteria, pathogens, virus can do that, infect SARS-CoV-2, and protein aggregates. Those can mediate neurological disorders. For example, this is my colleague Chen Yizhuang. She's an expert in Huntington disease. In Huntington disease, mutant Huntington is a problem, can cause the neurodegenerative disease. In this case, it's microglia. She showed that the mutant Huntington can cause lysosome damage, and then galectin-3 accumulate around damaged lysosome, and then lead to inflammasome activation. So then the other particulates. Now, if you work with nanoparticle, the researcher now know that if when you see galectins accumulate around endosome that they engulf, that they contain nanoparticles, the insufficient nanoparticle already got into cytosol. And another important factor that causes lysosome damage is oxidative stress, which is very highly implicated in various diseases. So I believe that this phenomenon of endolysosome damage, recognition of this damage by galactins are novel mechanisms mediated by cytosolically exposed glycans and lead to diverse cellular response and subsequently innate immunity and inflammation and diseases. Okay. So I think I was going to make this the last slide, although I have a few more slides. So here show that we have intracellular function of galactins and can be independent of glycan recognition or dependent on glycan recognition. So it may seem complicated, but I think in general, in a state of homeostasis, galactins can engage in cellular function through interacting with protein mediators. But when there's dangers, when there's a homeostasis altering situation, when organelles get damaged, then they can engage in glycan sensing process. So which I think is not complicated. I think what is challenging is outside the cell. What's their function? They are hundreds of papers already, but I don't think we know. I cannot say this publicly because I offend people because so many papers published, but indeed we do not know what they do. Because showing that they bind to this protein, bind to this cell type, this is not the answer. This requires some sophisticated approach to reveal their function, but I think it's worthwhile. They might be doing something very important, but we truly do not know. Okay, I think I stop here. Thank you.
Thank you for Professor Liu's the nice talk. So because we are out of the time, so can we have one short question in the anyone want to ask the question, or even in the online? No? Okay. Maybe I have a quick question. It's very similar to like the beta amyloid. In the past, we only think about the beta amyloid, the plug, only exists in the extracellular.
Right.
And right now, we find out the beta amyloid can produce in the intracellular and have the similar bad function.
Right.
But in the glycan, somehow in the intracellular function and the extracellular function may be totally different.
Right.
So it's maybe more amazing and interesting on that. So do you have any thoughts or comment about it?
Yes, yeah, thank you. It's a very good question. So first on the amyloid. Indeed, every time I have conversation with neurologists, those working on neurodegeneration, I ask whether those amyloid, those are intracellular or extracellular. Now it's clear, for example, mutant Huntington is intracellular, and now the tauophagy and this phosphorylated tau, whatever they form aggregates, they can inside intracellular. They can go outside, but they are intracellular aggregates, and they can cause damage. So this actually then the glycan become pretty relevant in those cases, like mutant Huntington induced neurodegeneration or tau aggregated tau. So that's clear. So in terms of glycan, indeed intracellular function, extracellular function, I think is quite different. So glycan-dependent function intracellularly it's outside the cell, I believe, mostly glycan-dependent. Should be glycan-dependent. Inside the cell, if it's glycan-dependent, then they're using the same CRT. But outside the cell, of course, certain this endosome, lysosome damage inside the cell, this is not occurring outside. It's mainly intracellular phenomenon, right? So outside, it still can bind so many different things, so many different cells. Okay? If give me any cells, I think glycan will, one or two of glycans will bind to that. And related to beta amyloid, for example, it has been shown that the glycan-3 is accumulated around the amyloid plaque, but I do not know what the meaning of that. I don't know what it means, because it has not been related to any alteration in the function. But thinking about glycan as a lectin, as a Ningji-Su, I think they're probably very good in causing aggregation, right? Aggregation, cell-cell aggregation or protein-protein aggregation. So that requires some approach to know when they go out. It's not when you inject glycans, but when the natural occurring endogenous glycan, when they leave cells, where they go to. When they go to, where located. And are they engaged in cell-cell interaction or forming aggregation of cells or proteins, right? Yeah. So, yeah. Thank you for a very important question. Yeah.
Thank you so much. 他说另外一个会议他还是没有办法离开，所以请我带致赠结缘品给我们院士。
Okay. All right. Thank you. 谢谢。Okay. All right. Okay.
谢谢。
谢谢。
好，那。